Thank you to our audience online. Thank you for joining us. If you're wondering, no, I am not Debbie. My name is Beth Orcutt. I'm our Vice President for Research. Deb is unfortunately not feeling great today, so I'm filling in to welcome you all to our second Café Sci for the season. It's such a pleasure to have you all with us today, as well as online, to learn about some of our science and what we're doing here. In case you've never been to Bigelow Laboratory before, or this is your first Café Sci with us online, just briefly, we are a nonprofit research institute based here in East Proof Bay. We're 49 years old. It's amazing to believe. And we really love to study the foundations of ocean health, how microbes in the environment perform important processes, the base of the food web, and really think not only how are those processes important for ocean health, but also for human health. And how can we use the knowledge that we have to better our experience on this planet together. As a nonprofit research institute, our discoveries, our solutions, the inspiration we have, it's all made possible because of the support of people like you and our supporters here in the audience. And I want to start today by thanking everyone that's a donor, that's a board member, that's an advisory board member, for the support that you give to the science and the education that we do here at the laboratory. Before I come to what we're going to talk about today, I want to also thank HM Pacer, which is a Maine-based financial advisor, for their continuing support of this Café Sci series. We're very, very thankful for the support that they give, allowing us to provide this kind of great programming for all of you. Sorry, I got my notes here. There's two things. For those of you in the room, as well as our audience online, as we go through the presentations, we'll take questions at the end of those. For those of you in the room, you can raise your hand. We'll bring a microphone around. For those of you online, you can use the Q&A button in the Zoom room to type in your question, and we'll try to get to as many of those as we can. Please refrain from asking questions during the presentation and hold them to those breaks when we have that time. So I'm really excited about today's presentation, which has the theme of science from a seed, growing bold science from big ideas. Launching big ideas often takes a little bit of a kickstart, a little bit of funding to kind of give you that proof of concept. Is this worth doing? Often, for instance, if we want to attract more funding from a federal agency or from anybody, we need to generate some of that preliminary data. And starting in 2015, with a generous donation of Mary Spencer, we started our very first formal seed funding program called the SASH-A, the Mary M. Spencer Entrepreneurial Fund. And this fund was created to allow our scientists to kind of say, I have this idea and I think it might work, but I just need to spend a little bit of time in the lab generating that preliminary data. And in the few years that we've been running this relatively modest seed fund, we've seen amazing returns. For example, one of our projects that started in 2017 with a couple $1,000 is now turned into a $27 million project that we're working on with collaborators. And we're going to, one of the scientists involved in that is going to be talking about that project in our Café Sci series on August 15th. And so these seed funds, these Kickstarter funds, are incredibly important to help us kind of get those initial ideas and take them to the next level. The projects you're going to hear about today are some early phase projects that we're excited. What's going to be the future of these ideas? So I'm really excited to hear from our speakers today, some of our newest senior scientists that are going to be speaking with us. We're going to hear first from Dr. Manoj Kamalanathan, who joined us a little over a year ago. I'll be talking about a project he has looking at the promise of soil algae. And then we'll hear a presentation by Drs. Catherine Mitchell and Dr. Julia Brown about a project they're working on collaboratively together, bringing totally different disciplines together to think about our oceans. So I hope you're excited to hear about these new ideas with us. We'll hear about 20 minutes from Manoj, and then about 20 minutes from Julia and Kath, with a little bit of a break in between for questions. So with that, Manoj, I think you're up.
Hi, can you hear me okay? Cool. All right. So yeah, I'm Manoj. I joined last April. My expertise is algae physiology and biochemistry. Today I'm going to talk about using soil algae as living fertilizer. So this connects Home Depot to Bigelow Labs to climate change, and I'll tell you how it works. So let me start by this graph. So this is a map of our planet, of course, and you can see that there are different colors here, and it talks about the state of the soil across the planet. And yellow means the soil is stable. Orange means it's degraded. Red means it's very degraded. So you can look at this graph and you can say Canada and Russia are the only places that are doing pretty well. But the rest of the planet are having either degraded or very degraded soil. The other thing I wanted to show you guys is a pie chart of greenhouse gas emissions that are responsible for climate change. And that pie chart shows the top 5 sources of greenhouse gas emissions in our planet. And agriculture is the top 5. So what this tells us is these two graphs tells us that our current way of current practice of agriculture is causing issues. It's degrading the health of the soil, and it's also accelerating climate change. And if you look forward into the future, like our population is expected to grow to 10 billion by 2050, which means we need to produce more food, like 56% more food, which means we have to do like more agriculture at a faster pace, which means those two problems are just going to get worse with time, right? So we need to find some solution. We can't just continue this way. And we think that algae can have some promise here. So just to give you a very simple overview of what algae are capable of doing, here is a simple cartoon of the photosynthesis. So they take in energy from sunlight, and they use that energy to bind the elements of CO2 and water to make oxygen and sugar. And that's photosynthesis. And they've been doing this for quite some time. So if you look here, that's zeros right now. And if you go all the way, that's billions of years into the past. And in the y-axis, in black, that's CO2, and in blue, that's oxygen levels in our planet. Up here, it's all the organisms that started evolving on our planet at what time. So it's cyanobacteria, then algae, and then the plants came later. So you can see the level of CO2 began to drop and oxygen started to increase. So cyanobacteria and algae have been playing a big role in making our planet what it looks like today. So they're very powerful in doing what they do. So when we think of algae, we always imagine them growing in water. Like here's a satellite image of the Baltic Sea, and all the green stuff we see there is algae. Kat and Julia will be talking about the power of using satellite, which is after me. And here's a picture of Cross Lake, which is up in northern Maine, and that all the green stuff you see is algae. So they grow very well in water, but they also grow in soil, which is mostly ignored. Sorry. So here's an example of algae growing on soil, and that's a pepper plant. And that's where the Home Depot story comes in. Okay, so that's a stable biofilm green layer up there. So I'm originally from India, so I like spicy food. And I moved to Maine, so it's hard to find spicy food if you have noticed. So I decided to take matters into my own hand and grow pepper plants at home. And that's where I went to Home Depot and I saw this green layer of algae growing. So it's like, oh, let's grow it. And let's take it in the lab and look what's in there. That's the soil algae in liquid medium. Wait a second. Yeah, so that's these are the different things I saw in the microscope. So that's a green algae, that's a Taro, that's another green algae. It's again a different green algae. And that's a cyanobacteria. So all these guys they all contain carbon because CO2 from the atmosphere. But these guys are a little more special because you see this, spindle-like structures. They are heterocysts, so they're capable of fixing atmospheric nitrogen too. So they can convert nitrogen from the atmosphere into proteins. Okay, so all of them are present in the soil algae. So and if you and then I started went back into the computer and looked at some literature, and then I saw that soil algae you can see if you go for a hike in the ground, okay, and you can see all these different kinds of habitats, and they contribute to they capture up to 3.6 gigatons of carbon from the atmosphere, which is quite a lot. It accounts for a total of 6% of primary productivity, which means capturing CO2 and converting into biomass. I was, and then we look at the total land area we use for agriculture. So almost 46% of the habitable land is used for agriculture, which is a big chunk. So if you combine the power of soil algae capturing carbon from the atmosphere with the total amount of land we use for agriculture, we can actually increase the amount of carbon being captured if you intentionally apply soil algae in agriculture. So that is the idea. So we decided to test this with the help of Spencer Fund in our lab. So that's where the lab comes in and climate change comes in together with Home Depot. So here is a cartoon of the current way of agricultural practice. That's an imaginary apple orchard, and with an imaginary farmer spraying imaginary fertilizer. So what's happening here is all the plants are taking up the CO2 from the atmosphere. The farmer usually tend to apply excess fertilizer than what's needed. The plants take up a large portion of this fertilizer, but a lot of it is washed away into the waterways when there is rainfall. And that excess fertilizer then causes algal blooms and kill off. We call it eutrophication. And overall, the quality of the soil is also lower in this case. But our hypothesis is if we add algae, what could happen is the soil algae can uptake more CO2 in addition to what the plants are already taking up. They're also very good at taking up nutrients. So if the farmer is applying excess fertilizer, they can take whatever is not taken up by the plants. So when there is a rainfall event, there is less of fertilizer being washed away, entering the waterways. So there will be less algal blooms. And the other thing is when they fix carbon and uptake all the nutrients from the fertilizer, they convert into organic form, and they will return it back into the soil. So they increase the organic content of the soil, therefore increasing the health of the soil. So it would help with the first graph I showed you about the yellow, orange, and red part. Okay. So overall, we can capture CO2. We can enhance soil's organic carbon and nitrogen content, and especially if there are cyanobacteria, we can bind to atmospheric nitrogen. So, which means the farmer won't have to apply nitrogen in the form of fertilizer, or not as much at least. And it can retain excess fertilizer in the soil. And because the farmer is reducing their carbon footprint by capturing more CO2, the farmers can participate in the carbon economy and gain carbon credits and make profits. And overall, this idea is super simple and very cost effective to scale it up in a global level. So right now we're testing, this idea is very new, there's not much out there in the literature. So we're starting from ground zero. So right now we have a summer intern, Ellie Strange. She's from South Carolina. She's testing, asking very simple questions using photograph on the left right there about her experimental setup. So I'm going to present a couple of slides about what she found here. So first thing we wanted to ask was, if, soil algae grows naturally, but if we want to intentionally make them grow on a farm, is it possible? Because, when things happen naturally, but we want to do it and it doesn't happen. So this is quite common in the lab. So we tested different levels, different amount of soil algae. Like 1 ml, 2 ml, 2 ml, 5 ml, and control being 0. And then we monitored the growth of algae on the soil. And what we found here is control showed little to no growth of algae because we didn't apply. And then in the other treatments, we saw almost no change, but there was a lot of algae, which tells us that as little as 1 mil is enough to start a stable biofilm in the farm. We can probably go lower. We just haven't tested. So we thought 1 ml was the smallest volume, but we can go lower. So if we want to do it in the farm, it is possible to start a biofilm. And then the next question we asked is there any negative impacts of this idea of using soil algae? One thing that comes to mind is, like I said, fertilizer being washed off and causing algal blooms. These soil algae can be washed off, and it can be a source of algal bloom, right? So we wanted to see if that's possible. Short answer is no. Because right here, you're saying in this flask is also soil algae, but they're dead. And this one is soil algae very alive. The difference between them is there is no soil here, whereas this one has it. So I tried to grow the soil algae just like we grow all the other algae from the oceans. And they didn't grow because they wanted something from the soil. The soil is giving some unknown chemicals, which we haven't found what it is. So when I give them, they're very happy. So the way we tested how much of that unknown chemical they need is by this setup. So we used the soil, known quantity of soil, put it with hot water and in a French press, just like you make coffee. And we made a soil coffee so there. And then right here, so we added different amount of that soil coffee, and we measured the growth of algae. And we saw a linear increase. The more we added the soil extract, the more we started to see growth in soil algae. So it was proportional. But this graph also shows that without it, it doesn't grow. So in case of a rainfall event, this soil algae entering the waterways is not going to cause any bloom. So going further, we have a few more, actually a lot more questions to ask, but here's 4 questions I chose to put in the slide. One is how much carbon do they absorb? So we want to measure the rate of carbon they take up from the atmosphere and how much is actually stored into the soil. Second question is how much excess fertilizer will they retain in the soil and in what form? Third is how often do they make it available to the plants? Because if they are stored inside the cell and not released it back in the soil, that means it's not available to plants, right? So we want to see what's the turnover. And the last is, do they have any negative or positive or no effect at all on the plants they're growing with? So any sort of synergy or not. Yeah, so thank you for your time. If you have any questions?
Thank you so much, Manoj. Do we have any questions in the audience for Manoj about this? Yes, right here.
Have you thought at all of working in collaboration with this process with the gardens, the Coastal Maine Botanical Gardens, in relation to fertilizing their gardens?
Yes, so this is an early phase idea, and it's using live organisms. So before we put it out there, we want to test it in the greenhouse control facility. We are actually working closely with Maine Department of Agriculture to figure what's the best pathway to get there. The eventual goal is to work with the farmers. Yes.
Question here.
You mentioned biofilm several times, and it made me think that if you simply put the algae into the soil and mix it uniformly, only the surface layer will see the sun. Is that correct interpretation?
Yes, so that's correct. So we're not going to mix it. We're just going to spray it on the surface because we want them to grow. They need the sunlight to grow.
Here's a better question. What ingredients in soil cause the algae to do its thing?
That we don't know because there's literally thousands of chemicals. I can spend my entire lifetime testing one of them, but I would like to explore that as well. There's a few top candidates we would like to test, but right now we don't know.
Before we go to another question in the audience, do we have any questions online? Okay, great. Well,
Good stuff, Manoj. Do you know what the durability of the soil algae is? How long lasting they would be?
So the graph I showed you with different amount, we waited until 3 weeks and it was still there. It was actually spreading more. So it's like a living system now. So we can keep them alive as long as we want, I guess.
Okay. A question here in the audience.
At the beginning, you talked about soil degradation. Could you tell us what you mean by soil degradation and how the algae would change that?
Okay, so that's a very good question. So because soil degradation happens when you grow like overcrop in a particular soil, and you totally rip the soil of all the nutrients and everything it can give to the plant. One thing we want to try is there are a lot of abandoned farms out there. So we want to take a soil from an abandoned farm, add the algae, and see if it can revitalize that soil. So that's one of the questions we want to ask in future.
We have a question back here, Manoj.
Hi. Okay, wait. I don't know if this is loud enough, but okay. Are you worried at all that when you introduce this algae into the wild, that it's going to cause competition between native microbes and the algae itself? Or is there no competition at all, or have you not looked yet?
Okay, that's another good question. So the experiments we're doing in the lab has natural bacteria in the soil. So far, we haven't seen any negative impacts of adding on the plant. I don't know, we haven't looked from a soil microbial perspective yet, though. So we have to look at that.
Time, all right. 2 more questions, and then we'll move on to the next one.
Is it possible to capture aquatic algae for use in soil applications, or are they completely separate species?
So they are different because I tried to grow them like aquatic species and they didn't. And the other thing is people have used aquatic algae, but they usually dried and killed it and applied it just like powdered fertilizer, not in its living form. And that's where this stuff that already grows in soil would be a better fit. Yes.
All right, last question, we get Manoj.
You mentioned cyanobacteria.
Yes.
I'm wondering how that fits into what you're doing. Is the cyanobacteria present in or on the soil, or is it part of what you're adding with the algae?
So it's already part of the consortia. The stuff I isolated from Home Depot, it was already in there. And cyanobacteria is just like algae, it can photosynthesize, but this one can also fix nitrogen.
Do they complement each other? Like, do the cyanobacteria do better with the algae and the algae with the cyanobacteria?
Yeah, so they all are coexisting together. So it's like a family almost. So far, if one was competing against everyone, then you would eventually what we would see is just one kind of algae with time. This has been like a multicultural consortia for at this point over 9 months. So it's been pretty stable.
All right, please join me in thanking Manoj for sharing his idea. All right, and keep coming back to our Café Sci to hear how this project evolves. I'm going to hand this over to, oh, you're going to use that. Okay, great. I'll put this away. So coming up next, we have, we're going to switch gears here a little bit, although Manoj gave a nice little preview. We're going to hear from Drs. Catherine Mitchell and Dr. Julia Brown about their idea of looking at big data approaches from different lenses, combining satellite data with microbe genome data to learn about our environment. So I'm not sure, Kathy, you starting first? Okay, then I will let you just take over. Here's one.
Great, thank you.
Yeah,
I think, yeah. Do we have to use one, or is it going to be?
Use the black one. This one.
Okay. Oh, thank you. Okay, we're good. Okay, hi everybody. So I know this talk has been advertised as mostly me, but actually you're getting me and a colleague instead. And so I'm Catherine. I'm a physicist looking at the oceans on a large scale.
And I'm Julia. I'm a microbiologist, and I look at the ocean on a very microscopic scale.
And we're here today telling you about this project together because it's really a story about what happens when you put two people that do very different science in the same room. And so, like, yes, Julie and I have both studied the ocean, and we work with lots of complex data doing computational research. So we spend most of our time doing our work on our computers rather than doing experiments in the lab or going out in the field on a ship. But we do it from very different viewpoints. I use satellites to look at the ocean, and Julia looks at the microbes in the ocean almost individually. So for a few years now, we and some of our colleagues have been meeting on a Friday morning for a coffee to talk about computer-based research that we've been doing. And it's been a fun group. We have a lot of good conversations, but something we speak about a lot is how we could try and do some projects that cross some of the boundaries within oceanography, like the types of work that we do. And Bigglow's seed funds really gave us the opportunity to try and do that. And so this talk is really about what happens when you put us in the same room. And what emerged was the question, like, what can satellites tell us about microbes in the ocean? So I'm going to hand it over to Julia to talk about those microbes.
Thanks, Kath. So when I think about and study the ocean, an image like this is what comes to mind. And this image is what really got me into the field at the beginning. It blew my mind. I'm trying to get that little guy out of the way. Sorry. Okay, so what you're seeing here is a sample from Booth Bay Harbor. This is 2 milliliters of seawater stained with a stain that when it associates with DNA, it fluoresces when you look at it under a microscope. So the big dots in this image are bacteria, and the small sort of distant dots in this image are viruses. And they're both really important in the ocean. And anywhere you sample the ocean, you're going to see an image like this if you create this type of slide. And in my research, I'm generally interested in how these two groups interact. But today's project focuses on the bacteria, which are present in concentrations on the orders of millions per teaspoon of seawater. These bacteria, which I'm calling bacteria plankton in this image, they're sort of very small plankton that float around the ocean, sit within a key loop within the marine carbon cycle, which I'm showing here. It starts with CO2 that comes in from the atmosphere and dissolves in the ocean. And that CO2 can be taken up by phytoplankton, which using photosynthesis convert that CO2 into organic matter. Which some of which can be taken up by the bacteria plankton. All along this cycle, there are moments when there's death or predation that causes some of this carbon to be taken out of the cycle, and it will either sink down deep or move into higher organisms as it gets eaten up. And so the question is, what is the balance of carbon in these systems and in this cycle? How much carbon dioxide that is used by photosynthesis gets stored and sequestered within the ocean? This question is critical to understanding our current and future global ocean and atmosphere. This is not all too different from systems on land. So maybe a more approachable way to think about this cycle is to think about a forest. So in a forest, you've got trees and underbrush that are photosynthesizing, and they're creating organic matter in the form of leaves and acorns. And then you have things that are consuming that organic matter, like mushrooms, like poorly drawn chipmunks, as well as bacteria. And you can imagine that if the photosynthesizers in this image, the trees, like if we move, instead of a forest, we go to a grassland, that the types of carbon that's generated that then gets eaten up by consumers changes, and those consumers themselves change. You have different consumers in that system. And so it's the same in the ocean. The consortium of phytoplankton that are there affects what types of organic matter is produced, and in turn, what types of bacteria respond and consume that organic matter. And so one consortium might activate or select for a certain group of bacteria, while another consortium might select for other bacteria. And so to identify microbes and understand what they're doing, I don't look at images like this that much anymore. Instead, I look at something like this. I look at raw data, A's, T's, C's, and G's that represent the genetic material extracted from microbes in the water. These data come from hundreds of different types of cells that are all finely tuned to specific conditions in the environment. From there, you can think of me as taking the role of kind of a crime scene forensic investigator. The suspects are microbes from the global ocean. And by comparing DNA DNA from samples to a database of known ocean microbes, I can figure out the identities and behavior, more or less, of these suspects. But remember, there are millions of microbes per teaspoon of seawater. So the scale of each sample is huge, and I'm looking at an equivalent of maybe all human crimes happening at the same time in every sample. Like there's a lot, this is big data we're working with. It's not, and this data is not easy to collect. It requires going out into the ocean on research vessels and filtering water. So there's not a lot of collections of information on microbial populations that are big enough and span the globe enough that we can really figure out global trends and what they're doing. So we need, or we're thinking about ways in which we can learn about microbes without having to go on these huge expeditions all the time. And that's where Kath comes in.
Thank you. Great. So what Julie has been talking about is the down here, the DNA, the microbes. And what I do is I'm thinking like way up here. I'm using satellites to look at the ocean. And when I first learned that you could use satellites to look at the planet rather than just using satellites to look into outer space, I thought this was really cool. And I wanted to know more. And 8 years later, here I am, like still using satellites to look at the ocean. And one of the great things I think about using satellites is we get this daily view of the ocean, of the whole planet. And so we can really see what's happening on the global scale, like Julia was just talking about, not just on our doorstep or where we happen to be sampling. And so that means we can then begin to answer questions like how carbon is moving through the ocean on a global scale. And so these satellites that are looking at the ocean are really looking at ocean color. And by that, I really do mean the different color of water. And Manoj actually mentioned this in his presentation and showed us a cool slide. And like what we're looking at here, maybe people recognize this scene. I'm seeing a few nods in the audience here. So this is, I'll put the star on, or the arrow. This is where we are right now in the lab. This is the Gulf of Maine off of the coast out of the Dam-Resculptor River. And what you can hopefully see, I added a line on to, in case the colors weren't too clear in this image, is these kind of two very contrasting regions in this image and those different colors. And so you've got those blues, and then you've got a more like turquoisey, greeny, yellowy color. And as Manoj mentioned, when there's phytoplankton or algae in the water, it makes it a kind of greener color. That color change is significant enough that can be detected by satellite. But there's some other material in there that can also affect the color of the water. And it's some of that organic matter that Julia was talking about. Some of that is colored and will make the water look a kind of yellow color. And so I think of that like tea. So tea is just dried, dead plant leaves steeped in water, and it makes that nice tea color. And so the same thing happens in the ocean when you get dry, dead leaves from the land, and with the rain and that material seeps into the rivers and out into the oceans, it will color the water a bit. That happens quite a lot in the Gulf of Maine. But also those ocean microbes themselves, when they die, they will color the water a bit themselves as well. So we have all these different things affecting the color of the overall color of the water, and that is what is seen by satellite. And so here, this looks like we're looking at a nice photograph, but really the satellite data is a little bit more complex than that. So we're going to zoom in on this tiny little spot, and what satellite data looks like, if you zoom right in, is a bunch of pixels, this grid of little boxes of information. So a satellite is like a computer screen or your TV screen or your phone screen. It's made up of a bunch of lots of little individual pixels. And each pixel gives you one bit of information, one data point. But an ocean color satellite doesn't just provide one grid of pixels, it provides much more. And each of those grids represents a different color. And so to demonstrate this, for this image we've been looking at, zoom out again. So like we get this red grid, and then we get the yellow grid, and then we go through into the greens and into the blue and into purples and ultraviolet. And so here we end up with, for this satellite, 7 grids. That will vary a bit depending on the satellite, but within these grids for this full satellite image, we have about 4,500 by about 3,000 pixels. So about 13.5 million pixels in one satellite image. in one of these grids. And so times that by 7, you get up to about 950 million pixels for one satellite image. So again, like we're getting into these realms with working with a lot of data and handling a lot of data. And so normally what I do is try and use all these different colored grids to figure out what phytoplankton are in the water, where they are, when they're there. But with this project, we wanted to take it a step further and ask the question, can we predict the activity of microbes from space using satellites? And by that, we're really just saying like what microbes, what bacterial plankton that Julia was talking about, like what ones are there and what are they doing? And so how do we go about trying to combine these two very different worlds? So we made use of this really, really cool data set. It's from the Taro Oceans mission. And so Taro is a schooner that does science. It's a floating laboratory. And so it's a 2-masted schooner. It's about 118 foot long. It was designed for polar waters. You can see a couple images of it in the bottom of the screen there. And so in 2004, it did its first science voyage up to Greenland, then a few years later to the Arctic. But from 2009 to 2013 was the Taro Oceans expedition. And so that was 4 years. And it went, you can see where it went, this red line on this map that kind of goes all over the place. And so it's going through the Pacific, the Atlantic, and the Indian Ocean. So it covered a lot of ground, a lot of different places in those 4 years. And all those little dots and squares on that map are just the locations where the ship did some science. And so there's 210 of those spread out over the planet. And what is really unique about Tara, apart from the fact it's a sailing boat that does science, is that it was one of the first science expeditions that measured ocean color while on the ship, as well as measuring sampling for omics. So taking some of those DNA measurements that Julia needs to do the science that she does. More recently, there's been a few more cruises and expeditions that have been starting to gather this type of data, and some of that data is starting to become available. But we focused on this data set because it was really the first one, and the data is already there and available, and we could kind of get our hands on it and start looking at it. So I'm going to hand it back to Julia to talk about what we found. Great.
Okay, so the first thing we did was to identify sites and track down data where they collected both information on ocean color, as well as what microbes were there and what they might be doing. And this resulted in 45 of those sites having all of those data sets available to us. And they were globally distributed. We found sites in the Pacific, the Atlantic, the Indian Ocean, as well as some in the Red Sea. So after downloading this data, doing a lot of processing and quality control, we began to calculate the relatedness between these different sites based on these 3 different criteria. And so from here, I'm going to start to show you some of these results. So first we looked at ocean color and we said, what sites look similar based on ocean color? And so that's what this result is showing you. So now points on this map are colored based on the clusters or the shared similarity between sites. So if they're the same color, it means if these points are the same color, it means ocean color looks similar. And the takeaway here is that there are a lot of sites that were globally distributed that had similar color. And you can see that with the sort of turquoise points that you see in the Pacific, Atlantic, Indian Ocean, and even in the Red Sea. There are a couple of sites, though, that we found to be unique and looked totally, well, not totally unique, but they looked different than the other sites that we saw, including this one site on the Patagonian Shelf, for example. So next, I'm going to change the shape of these points according to how similar they look to each other based on what microbes were present in the samples. And so once again, points that are the same shape are sites on the map where similar microbes were present across those points. And so For example, the circles on this map are once again globally distributed. We see that very similar microbes were found at across the global ocean. Once again, just 2 sites stuck out as being completely unique relative to the others. One of them being this Patagonian Shelf site that we'd seen before. And so finally, we wanted to look at what those microbes were doing. They might be sitting in the water, but are they active? What processes were they involved in? And we focused specifically on genes that were interacting with carbon. Since ocean color is sort of an indicator of who's in the water and what types of carbon are in the water. And so when we looked at compared sites based on that particular set of genes, we saw that for the most part, the same types of genes were being expressed all over the ocean, save for one site, right there on the Patagonian Shelf. So why this site? Why is this particular spot unique relative to all the others that were sampled by Taro? And to sort of think about that, we're going to show now a map that just shows the distribution of phytoplankton across the ocean on sort of an annual scale, or sort of averaged annually. And so in this figure, the purple and blue colors are very low phytoplankton, while the red colors mean that there was a lot of phytoplankton in the water. And most of the sites that Taro sampled fell into locations where they were mostly blue, purple, and maybe a little light green. But this Patagonian Shelf site lights up. This is sitting on the continental shelf, and it's a region that's very dynamic and experiences blooms of algae called coccolithophores. And it's showing us that we can see a signal when it, in some cases. And so this one little site gives us hope that this technique of connecting ocean microbes and the things that they're doing to ocean color might be something that would be useful. But we need to sample more dynamic places. We need to sample places that have different colors. And so lucky for us, more and more data is coming online that has information about microbial data and ocean color. On the left is a map of sort of all the sort of global sampling expeditions that have looked at microbial activity and microbial communities. And although a lot of these sites probably didn't have optics data or ocean color data sampled at the same time, because there are a lot of samples out there, we might be able to figure out the ocean color at a subset of these time points, and that can help us better hone our technique.
And the thing that I'm really excited about is the up and coming deployment of PACE, which is the new NASA satellite that's going to be looking at ocean color. It's going to be launched very early next year, and it's going to provide a much higher resolution of the ocean in terms of how many colors that it can see. So if we think back to those different colors that I showed you earlier, there were 7 different colored grids, and that's what's standard right now for all ocean color satellites. But PACE is going to give us almost 100. And so that's a lot more information that we'll be able to use to pull apart some smaller differences than we're currently able to see in our satellite data. And which I think will be much more useful to help look at some of these questions. It's like looking at the microbes and what they're doing. And so we are thinking about where we can take this next, both by expanding our analysis to some of like these up and coming and existing data sets that are now coming online. But also we've been talking a lot about doing some data collection ourselves and like where we would go to get this nice range of dynamic, different conditions that we would need to be able to kind of dig into this idea a little bit more. And that is all that we have for you. Thank you. Great.
Thank you so much, Kath and Julia. Are there questions here in the audience and or online? Yeah, but actually there's a question back there, but go ahead. If there's one online, go ahead.
Can you talk more about with PACE, how that might expand this work or make other work possible, or how you think it might make this kind of work possible?
Yeah, sure. Thanks for that. So with paste, like I said, it's going to have these almost 100 colors, and so it's a lot more information. And so we need to start thinking about different ways of using that information. And right now, the ocean color field has been doing their techniques in one particular way, but I think where we need to start moving is into more of the machine learning type approaches, because we're suddenly getting so much more information that we need to handle and deal with. And when you have 7 wavelengths, it feels like a lot to work with, but when you suddenly have 100, it's going to be, we can't keep doing it the way we've been doing it all this time. So it's exciting. And there's a lot of people doing a lot of good work trying to figure out how we're going to interpret all that data.
All right, we got a question back here.
Hi. So my question is, when you're looking at ocean color, does it tell you any other statistics about what's happening in the ocean there, like in that particular place? For example, is there a high level of pollutant? Is there a higher level of salinity? Are you getting any of those other types of data points that could influence why it's a hot spot or why it isn't a hot spot? And that leads me into another question. Can you look at smaller bodies of water like lakes that could be polluted and things like that? Sorry, I love pollutants. They're so interesting.
It depends what the pollutant is and if it colors the water. So the satellites that I use look particularly at color. And so ultimately, if whatever you want to see affects the color and it affects it enough, then you should be able to detect it. To salinity, like, yes, there are satellites that look at salinity and temperature, but they do that in slightly different ways with slightly different satellites. So you can combine those observations to look at some of that. And a lot of people do that. To the question about lakes, yes, people look at lakes. Depends the size of lake you're interested in. Again, the standard ocean color satellites right now, they can't see anything smaller than 1 kilometer. So like great for the Great Lakes, not for like the Damascotta River. I know that's not a lake, but or like other smaller water bodies. The satellite image I actually showed you was actually a land-based satellite. It can see much smaller resolution, and you can see water with those. You don't have as many colors that are suitable for looking at the water. So there's all these trade-offs that exist depending on how high spatial resolution you want to see versus how high spectral resolution you want to see, because there's only so much data the satellite can acquire and then transmit back to the Earth. So yeah, people do use satellite and satellite ocean color to look at lakes. That's the short answer.
I think there was a question over here. There we go.
My question is not exactly on your work, but you showed a map there with data from, obviously there must have been hundreds or more of institutions that collected that data. How cooperative are all the various institutions in sharing that work? And when you started out looking at your map, how did you go out and collect all this stuff that must have been done by hundreds of other sites?
Do you want to do this? Well, I'll give an answer and then Julia can maybe answer something in. So the map that I showed with all those hundreds of sites they were all measured from one ship. It is a big cooperation that had multiple people involved in it, but it was one planned expedition that was coordinated by one body, and they collected that. All that data was collected from that sail ship. And then all that data was made publicly available. So scientists like us that we're not part of the data collection part can come along and use that data. And that's a big part of what we have to do as scientists, is when we get funding from federal agencies, we have to then make our data publicly available so that anyone can use it. It's not just for us, like taxpayers' money is what we're using to do our science. So we give back in terms of making our data available for other people to come along and do that. If that answers your question. Did you want to add anything?
Okay, question back here.
It's sort of a non-scientific question. I'm a summer person from Florida, and I'm really worried about the unexpected increase in temperature How do you account for that on a global basis, these dramatic increases in the temperature of the water? Does that mess up what the microbes are trying to do?
I think the short answer is we're still figuring that out. There's probably like changes, big swings in temperature are probably affecting sort of rates and processes that are happening. But I'm not sure we know exactly how that's going to go about.
We have another question back here.
With your 7 pigeonholes of color spectra and your sequencing data that you've done, have you begun to draw correlations between individual species of organisms, single cell sequencing data with the various wavelengths of the spectra that you're getting from your satellite images?
Yes. Yeah, we showed sort of the broad swath of this data, but we have the next sort of step and where we've begun to push this analysis is to look at so we see these trends, what genes are causing that particular site to be unique, what colors are causing that particular site to be unique. And even more broadly than that, we're clustering things, but there are obviously differences between every single site. And so we're looking at how, like, for example, all of the seven wavelengths that we're studying individually contribute to the variation in the microbial data.
Oh, we have a lot more questions. Okay, I'll come back to you, Barbara. There's one back here.
When we started out, they said you were trying to put big picture and little picture. I don't mean it that way, but the two things you did together, and that was the point of the seed project. What do you see as the future to come out of that seed? Was it worth your time? Are you going to go back in the labs and do different things, or what are you thinking?
We're thinking about how to move this forward. We've been talking about, lots of different possibilities. One is there's a gnats. You should talk about it, but there's this time series that goes across the Gulf of Maine that Kath is now working on, sort of projecting into the future. And if that were to be able to continue to happen, we can start to integrate microbial sampling into that expedition. And the Gulf of Maine is pretty dynamic, yes, color-wise. And so it would be a good place to sort of start to ground truth some of the trends that we see on the global scale, but like here in Maine. Yeah, and there are other ideas too, but we're also just sort of keeping our eye on opportunities and ways that we can think about, getting the science supported.
This would be, I guess, a different group of scientists, but would you be able to layer over marine animals over all these different colors?
Yes, and people do that. Depends to what level you're doing that. People do it on an individual basis. We have people at the lab that kind of work a little bit on that, but people also do it in terms of forecasting. So Nick Record that's at the lab, he does a lot of forecasting, particularly with the North Atlantic right whale. And so a lot of those forecasts that Nick does, he uses satellite ocean color data in those forecasts. And so that's a big part of what I do outside of this particular project, is like thinking about ways that we can get some kind of information from ocean color data that's useful for a lot of different reasons. And like that is one of the things that I'm thinking about, is what kind of thing can I get that is useful for looking at where mammals are and bigger things. Yeah.
Good. We had another question here.
Okay, the first half is for Julia. Do you take samples in the same place over different seasons so that you know that they're always the same?
Well, we wouldn't expect them to always be the same,
right,
over the seasons. But but yeah, I mean, we're using data that's currently available to us. And so we're not, we can't, with the data set that we have, we just have to, one thing that we need to consider that we haven't really integrated yet is time of year per site and how that relates to some of the patterns that we see.
Okay. Well, then the next half goes to Catherine, because if the pictures are being taken, I don't know how often, do they take them regularly, or do you just have one set?
Yeah, so we get a daily picture. So that is something we've also not done, is like check the time from the satellite image to the omics data. But that's because what we actually did in this project, and I didn't go into these details, was they measured ocean color on the ship. And so even though I was talking about satellites and satellite ocean color, and that's where this project will go, the ocean color data that we used in this project was taken on the ship at the same time exactly as these measurements that were made. So in this case, they match up perfectly.
With a camera?
No, with a fancy instrument that counts photons, but very similar.
Good. Do we have any other questions online? Okay. Well, please join me in thanking Kath and Julia. What I'd like to emphasize for all of these presentations and our presenters, these ideas are very new. They were kickstarted within less than a year. And so it's very brave of you as scientists to share your very early ideas with us. As a scientist, I usually like to wait till I know the full story before I tell others about it. So, but that's, again, part of what we're trying to do with these seed funds, both with our entrepreneurial fund that was started by Mary Spencer's donation, that has sponsored things like Manoj's project, which has a very applied goal in mind in terms of thinking, how can we take the science that we know and try to solve a problem that we have in the environment? And we wanted to actually replicate that model. And we started a new seed fund a few years ago to fund some of our basic foundational research. And that fund is actually sponsored by our annual fund. So any of you that are contributors to our annual fund, thank you for helping make this kind of work that Julia and Kath talked with us about today possible. So please join me again in thanking both Manoj, Julia, and Kath for their presentations.